Very good morning to everyone. Welcome to SEBA's expert seminar. This is number 3 for this year. So today we have Dr. Chua Ka Hui, one of our lecturers in SEBA, as a speaker. So let me briefly talk. Okay, so Dr. Chua is a lecturer in the SEBA of University of Malaya. So he completed his Bachelor of Science in Microbiology and PhD in Molecular Microbiology from So his research interest focuses on bacterial genomics and microbiome studies on the varieties of biological and environmental samples. So he has experience in taxonomic characterizations of novel bacterial species, and all those insect and also the environmental bacteria. So currently he is working on these plants AMF interactions and also the halotolerant plant growth promoting rhizobacteria from Malaysia's mangrove. So without further ado, so let's welcome Dr. Chua. And Dr. Chua, you can share your slide if you're ready. Okay.
Okay, can everyone see my slides?
Yes, we can.
Okay, so good morning, everyone. Welcome to SEBA Expert Seminar. I'm Dr. Chua, and today I'm going to present about a genome analysis on a bacteria, Pseudomonas, strain EB3, which is a halotolerant plant growth promoting bacterium that could alleviate the salt stress in banana plants. So plant growth promoting bacteria, PGPB, they are known for their ability to enhance plant growth under various abiotic and biotic stresses. This is due to their traits, including biological nitrogen fixation, phosphate solubilisation, production of siderophores, and so on. So it has been suggested that this plant growth promoting bacteria could be used as an alternative to chemical fertilizer that are harmful to the environment when they are overused. So this study will involve a bacteria from the genus Pseudomonas. Pseudomonas are gram-negative bacteria. They are known for their metabolic, versatile metabolism, and they are ubiquitous in the environment. Bacteria Pseudomonas could be both present as a rhizosphere and endophytic bacteria. So many species have shown ability to promote plant growth, and Pseudomonas are reported to produce antibiotics, bacteriocins that could help plants to cope with biotic stresses such as the pathogen attack, and they are known for their ability to produce siderophore, to perform phosphate solubilization and nitrogen fixation, which are actually plant growth promoting traits. So in this study, we are looking into the genome analysis of strain EB3. Strain EB3 is actually an endophytic bacterium which we isolated from a mangrove plant species, Avicennia alba. So this bacteria is halotolerant. It could survive in media with up to 600 millimolar of sea salt or 1,500 millimolar of sodium chloride. So strain EB3 demonstrated various plant growth promoting traits. However, the genomic and taxonomic information about this bacteria is limited, which is why the objective of this study are to perform whole genome sequencing and to determine the genome features of strain EB3, to determine the taxonomic placement of strain EB3, and to identify the genes of strain EB3 that could be involved in plant growth promoting. So to start with the whole genome sequencing of strain EB3, the total genomic DNA was extracted from the bacteria for a genomic library preparation. So the genomic library was prepared using a Nextera DNA sample prep kit from the Illumina, and the prepared library was then sequenced on a MySeq platform using a 2x250 pair end setting. So with the raw reads obtained from the sequencing, we first quality filter the raw reads using chemometrics before the novel genome assembly was done using the sulfate SPECT. So plasmid sequences were searched using the Plasmid Finder 2.0, and to assess the genome completeness, we have performed a Bosco analysis which search for the benchmarking universal single copy autologs in the genome. So as a result, a draft genome of strain EB3 was obtained. It is about 6.01 MB in size, and it has a 62.2% GC content. So although there are a total of 244 contigs in this draft genome, it actually scored a 99.8% completeness in Bosco analysis. So the genome has an average coverage of 108 times. There was no plasmid sequence detected. In total, there are 5,767 genes annotated, of which 5,545 are coding DNA sequences, 4 are rRNA sequences, and 73 are tRNA sequences. So to move on with the taxonomic study on strain EB3, we first perform a 16S rRNA gene PCR, and with the sequence obtained, we blast it against the EZ3 Cloud database and then closely related Pseudomonas species, which are closely related to strain EB3, are obtained for their 16S rRNA gene for a phylogenetic analysis. So we also did a genome annotation on the genome of strain EB3 using PROCA, and the core genes present in the genome of EB3 and the closely related bacterial species were identified using the software RORI, and these core genes were used for another phylogenetic analysis to determine the taxonomy of strain EB3. The whole genome sequence of strain EB3 was also used for calculation of overall genome relatedness indices, such as the average nucleotide identity of ANI using fast ANI, and in silico DNA hybridization using the genome-to-genome distance calculator. So as a result, 1,473 base pair 16S rRNA gene was obtained for strain EB3. And when we blast it against the database EZ3 Cloud, the top matches are Pseudomonas plecoglossidicider, which is sharing 99.86% identity with the 16S rRNA gene of strain EB3. And the second match is Pseudomonas juntendi, which is sharing 99.79% similarity with the gene 16S rRNA gene of strain EB3. However, based on a further phylogenetic analysis, it was shown that strain EB3 is actually distantly related from the top match Pseudomonas plecoglossidicider. Based on the maximum likelihood tree using 16S rRNA gene, it was shown that strain EB3 is closest to Pseudomonas alloputita KH7. So as you can see on the right-hand side, all of the nodes were obtained with low bootstrap value, and this indicates a low confidence in the results. Very often, we need a multi-local sequence typing of several housekeeping genes such as the RPOB, RPOD, and Carey D. Or we need to include more genes in the phylogenetic analysis, such as all the core genes in the genome, to obtain a complete picture of the taxonomy. So, which is why I've performed a core gene-based phylogenetic analysis, which involves 30 reference Pseudomonas species, which are closely related to strain EB3. This phylogenetic analysis involves a total of 1,497 core genes, and they form an alignment of about 1.56 MB. So the phylogenetic analysis revealed that strain EB3 is phylogenetically closest to Pseudomonas juntendi BML3. So as you can see on the right-hand side, each of the nodes on the tree has obtained with 80 or 100% bootstrap value. This indicates a high confidence in the result. So at the time of analysis, there are 23 Pseudomonas juntendi genome available in the NCBI database, of which 22 are patient-associated, and only one is an environmental strain from a water sample. So I have obtained all these genome of Pseudomonas juntendi from the NCBI database for another called gene-based phylogenetic analysis. So this genome share a total of 1,442 core genes, and as you can see on the right-hand side of the results, it forms all the Pseudomonas juntendi form a cluster in the phylogenetic tree, which includes strain EB3. So this result indicated high confidence in taxonomy placement of EB3 in the species Pseudomonas juntendi. So Pseudomonas juntendi is actually a relatively new Pseudomonas species. The type strain Pseudomonas juntendi BML3 was only isolated and characterized in year 2019 from a patient's sputum sample in Japan. But fortunately, this strain is not a multi-drug resistant strain, and there is little information on the other Pseudomonas juntendi. In our study, it is shown that strain EB3 will be the first report of Pseudomonas juntendi as a plant growth promoting bacterium. So we further our analysis into the calculation of average nucleotide identity analysis. This analysis, this calculation was done by comparing each pair of genome, and the pairwise value were obtained for a heat map, which is then showed a clustering of all the Pseudomonas juntendi, including EB3 in a cluster. So as a result, EB3 shared more than 97.7% NI value with all the other Pseudomonas juntendi, and it has lower than 88.4% with NI value with the other Pseudomonas species. The ANI species threshold for all bacterial species is actually more 55% or more. So this shows that EB3 actually fall in the species Pseudomonas juntendi. So another in silico DNA hybridization analysis, the same thing was done. We have obtained the pairwise value for each pair of the genome, and a heat map was created before a cluster was shown that a cluster was formed for all the Pseudomonas juntendi, including the strain EB3. So EB3 shared more than 82.5% DDH value with all the Pseudomonas juntendi, and it has less than 70% value with the other species. So the species threshold for this index is actually more than 70%, and both the ANI and DDH indicated a high genomic coherence at the species level with Pseudomonas juntendi in strain EB3. So further genome annotation was performed using RAS and EGG-NOG for functional annotation and assignment into functional categories. We also refer the prokaryotic genome annotation pipeline from the NCBI database for the annotation of this genome. The objective of this analysis is to identify the genes that are involved in salt tolerance as well as plant growth promoting traits in strain EB3. So this bar chart shows the distribution of gene in the genome of strain EB3 into different cluster of autologous group or COG functional category. As you can see on the right-hand side the graph, majority of the genes are assigned into category that are responsible for basic cellular function such as D, the cell cycle control and mitosis, J, the translation, K, transcription, and L, replication and repair. There are also a high number of genes with unknown function or unassigned into a category. As compared to the other closely related Pseudomonas juntendi strain, EB3 has a slightly higher gene in category L and M, which are the replication and repair, and cell wall, membrane, and envelope biogenesis. It retains about the same gene in category N and P, and similarly, there are a high number of unassigned genes in Pseudomonas juntendi strains. Overall, the result shows an even functional distribution of genes in the species. Based on RAS results, as you can see in the pie chart, the gene distribution of strain EB3 the major gene distribution was in the category amino acids and derivatives, carbohydrates, and protein metabolism. The other majority of the other genes are also assigned into categories which are responsible for basic cellular functions such as RNA, DNA metabolism, as well as protein metabolism. So compared to the other Pseudomonas juntendi strains, strain EB3 only shows slightly higher gene in the category nitrogen metabolism. It retains about the same number of genes as most strains for the other category, and an overall similar distribution, a functional distribution of genes in the species. So strain EB3 actually show a high salt tolerance. It survives in LP medium with up to 600 millimolar of sea salt and 1,500 millimolar of sodium chloride. So based on the genomic analysis, it was revealed that there are present of putative genes that are involved in environmental stress resistance, such as the osmoprotectant peptidase, the lipoprotein, aquaporin, potassium transporter, glycine betaine ABC transporter, the hydrogenase of betaine and aldehyde, as well as the other genes that are responsible for oxidative stresses, such as the catalase and superoxidase dismutase and glutathione transferase. Strain EB3 also shows the ability to produce siderophore on Cas-LB agar. From the genome analysis, we identified several genes that are responsible for ion uptake, such as the ferric ion uptake transcriptional regulator, ion ABC transporter permease, TON-B dependent siderophore receptors, and ion ABC transporter permease. Strain EB3 also show ability to colonize banana plant, as shown in the SEM image on the bottom. So putative genes that are involved in plant root colonization could be the peeling assembly protein, pre-peeling type N terminal cleavage methylation and domain-containing protein, pseudoporin, pre-peeling and acylin, as well as the other gene. So there are also putative genes identified in the genome of strain EB3 that could be involved in plant growth promotion, such as the IAA biosynthesis, or indole-3 glycerol phosphate synthase, indole-acetamide hydrolase, and there are genes responsible for nitrogen fixation, such as the neutral reductase, NADPH and nitro reductase, as well as genes responsible for phosphorus solubilization, such as the bifunctional phosphatase, metallophosphoesterase, phosphatase, other phosphatase protein, and in situ phosphate phosphatase. So all these genes are actually correlate to the ability of strain EB3 in performing phosphate solubilization, nitrogen fixation, and the production of IAA. So as conclusion, whole genome sequencing revealed the genome feature of strain EB3, and strain EB3 was determined as a species of Pseudomonas juntendi based on phylogenetic analysis, as well as calculation of various overall genome relatedness indices. Genome annotation has helped to identify various genes that could be related to the plant growth promoting traits of strain EB3. Here are my references, and thank you.
All right. Thank you, Dr. Chua, for your interesting presentations. So if anyone have any questions, so you can raise your hands or put your questions in the chat box, so I can read them out for Dr. Chua. Right. Anyone have any questions? Maybe I can just ask one first. Yeah. So what is the functions for this Pseudomonas, or in particularly what are the applications that we can use for future applications?
So these Pseudomonas, we have tested many of its plant growth promoting traits using in vitro method, and we have also done an in vivo study using banana tissue tissue culture pellets. So we then show that it has the ability to colonize the roots of banana plants. So we are hoping to develop this bacterium into a formulation which could be applied in the field and to reduce or even remove the needs for chemical fertilizer.
Okay. So can this Pseudomonas be applied to also other tropical crops as well, or just particularly for banana?
We believe it should be able to promote the growth of the other plant species as well. Right now, we are testing radish, which is, and then we have tested also on cucumber. And from the cucumber results, it was revealed that this bacteria promote root hairs in inoculated samples as compared to control, which is not inoculated. So in the future, we will test this bacteria on many more plants, and hopefully it should show a high versatility. I mean a versatile host.
Yeah. All right. Thank you. Is there any other questions from the floor? Anyone? Okay, so I think Dr. Aziza is asked. It's a comment, right? Thank you for your presentations. In the future research, I suggest that to use this bacteria as probiotic for animals and also the aquaculture food as natural fermentations, and we will expect to get the good results. Or is there any comment from Dr. Chua?
Thank you. And I think this is a good suggestion. So far, we have never thought of developing it into a probiotic for animals or aquaculture. But maybe it could be, we could try this. Well, try this out. Yeah.
Yeah. All right. Thank you.
Thank you, Abby.
Yeah, Dr. Nadia has raised her hand, so maybe Dr. Nadia, you can ask your questions.
Thank you, Dr. Chua, for your presentation. In your method of developing your phylogenetic tree, you use the co-gene based sequence, right? So is it a standard method to develop a phylogenetic tree and and especially for gene annotation? Because I'm not familiar with this co-gene. If you can explain a little bit, what are these core gene means?
Okay, actually the standard for bacterial taxonomic identification, right now it is still based on 16S rRNA gene. So for most of the bacterial genera, 16S rRNA gene alone is sufficient. But for bacterial genus such as Pseudomonas or other genera such as Escherichia, Bacillus, in which they contain a lot more species in the genus. 16S, a single gene such as 16S is not enough. So there are trends now asking for multiple housekeeping gene in addition to 16S, such as the RPOB or RNA polymerase subunit B, RPOD subunit D, gyrase, DNA gyrase, and so on. So overall, the trend is now to include more and more genes from the genome because of the availability of whole genome sequencing technology. And I believe in the future, it will be a standard as well for core genome to be included in phylogenetic taxonomy and phylogenetic analysis. So to answer your question, the core gene base is actually a collection of all the genes that are shared by all the genome of all species that you include in your analysis. So we, of course, we aim to include more gene into the analysis so that we get a robust result. But sometimes it might be difficult, like computing power is actually a limitation. Like in my case, I've included 1,400 genes, more than that, and I could only run a neighbor joining tree, to be honest, because of limitation in computing power. So right now there's still no standard in this approach, but I believe sooner or later there will be a guideline for this.
Okay, thanks, Dr. Chua. So, based on your previous experience, you also use 16S RNA for your gene annotation, and
Yeah, I would say 16S rRNA gene is a preliminary identification.
I see,
So it could
understand.
tell at least the genus of your bacteria. Like in this
Alright.
case, we can tell that it is a Pseudomonas species based on 16S, but exactly which species we can't tell. So the golden standard for 16S rRNA gene is that if your bacteria share 97 or 98% or higher with a reference strain, then your bacteria should be affiliated with that species. But in our case, this EB3, it is showing more than 97% with multiple Pseudomonas species, which is why it needs more than a 16S in phylogenetic and taxonomic study.
Okay, I have another question actually from your sample. So you found this EB3 strain, right? So did you find any other strain, or is it because this is the most abundant?
No. We actually found a collection of, I think, 400 bacteria, and we have tested first their salt tolerance because we are aiming to search for the halotolerant bacteria. So we filter them based on their ability to tolerate different salt concentration, and we've ended up with, I think a handful of bacteria which could survive in 400 millimolar of sea salt. So with this handful of bacteria, we further study their ability to promote plant growth, to colonize the plant roots, and so on. And we ended up with 2 bacteria, and EB3 is one of them.
Okay, thanks, Dr. Chua.
Thank you.
Okay, there's another one from Dr. Tang. So thank you for the informative presentations, Dr. Chua. May I know, have you done any lab work to prove those genes which were suspected to be involved in the tolerance to abiotic stress? And what is your future plan of wet lab research to further confirm the contributions of those genes?
So right now we have just studied the genome of this bacterium EB3. So we have planned actually to perform qPCR analysis on salinity treated EB3 and non-treated, so that we know exactly which gene are expressed under a high salt condition. With this gene identified, we could, in the future, we could use this gene in case we need another bacteria, which is more specific to a certain plant species. We could do a transgenic, or we could make a transgenic organism. And yes, we do have a future plan of wet lab, including to develop them into a formulation. So understanding the gene features of strain EB3 actually help us to foresee what could be the possible obstacles, and such as because, like I mentioned, most of the other strains, they are patient associated, and whether or not this bacteria could cause a disease, it is something we need to find out too. So these are basically our future plans on this bacteria.
Right, thank you. Is there any other questions from the participants? Okay, just a note to all participants. So in the chat box, we already posted the feedback form link. So you can just look at the chat box and click and fill up the forms. Right. So I don't see any further questions from the chat box. And yeah, so I think, yeah. So thank you very much again, Dr. Chua, for sharing your research with us. Yeah.
I've seen a
Yeah.
comment from Dr. Tang. Yeah, we could consider gene knockout experiment. Yeah. I would say there are a lot of genes we can first do maybe a transcriptome analysis to find out which are the genes that are highly expressed under salinity condition or even during plant growth promoting experiment. So those are the genes that we could target in a gene knockout experiment to prove that they actually work under those conditions.
Yeah. Okay, great.
Yeah. Thank you for the suggestion.
Okay, so without I think no further questions. So I think once again thank you, Dr. Chua, for sharing your research and interesting findings with us. So for all the participants, SEBA will occasionally organize expert seminars. So do keep in touch with us, or do stay tuned for the next expert seminars. So that's all for today's session. So thank you very much.
Thank you.